All right, good afternoon. Let's get started. You have a good weekend. Our exam is Friday, and I did post something on Friday, but just as a reminder, that information will start with chapter 15, which was kind of the epidemiological terms, the ways we describe the transmission of infection. Just a lot of terminology there with chapter 15 into innate immunity, adaptive immunity, vaccines, and what we're going to cover today on Staphylococcus. We will go on to Streptococcus today if we have time, but that will not be on Friday's exam. Staphylococcus in your book is in chapter 20, which, like I explained at the end of last lecture, the way your book does it, chapter 20 is infections of the skin and eyes. So a lot of the staph stuff is in 20. There's some strep stuff in there as well. There's the few questions that I think there's only 3 questions in section 20.2, bacterial infections of the skin and eyes. And one of them might be a strep question. So sorry, that kind of reached it. I couldn't do like one without at one time and one at another time. So just a couple questions there. So remember, homework is due at 1 o'clock on Friday as well. Okay. So we'll start staph today. Hopefully, maybe, I know we're having lab exams today and tomorrow that covers staph and strep. So maybe you'll pick something up from this that will help you if you haven't had your lab exam already. It's kind of nice as we start talking about our pathogens, try to build in kind of things that we talked about earlier in the semester, and you kind of get to see, oh yeah, there's an example of this or that. So it kind of hopefully helps kind of tie the semester together a little bit. So we'll be doing first important gram-positive pathogens. Start with the cocci. The major ones are staph and strep. Enterococcus is very closely related to Streptococcus. You inoculate an Enterococcus in lab here as part of your unknown. I'm not going to get into Enterococcus. We're mostly going to stick with staph and strep here in terms of our round pathogens here, round cells. All right, so Staphylococcus, there's actually, it depends how you count it, whether you count certain ones as subspecies or species, somewhere around 40 different Staphylococcus. So we're going to talk about the 3 that are most famous, and especially there's one that really dominates the clinical scene with Staphylococcus, so we'll really focus on one. But in general, this genus, they form grape-like clusters. So remember back early on in the semester, we talked about the different cell arrangements that we see for cocci, for round cells. Sometimes if they only divide in one plane and then stay stuck together, they form chains. Okay, strept, that means, that's I think Greek for chains. So the streptococci form chains. The staphylococci tend to divide, that cell divides in many different planes. So and then they stay stuck together. So we get these grape-like clusters. Okay, tetrads, only 2 different planes. Sarcina, a 3rd plane. So you get kind of cube-shaped arrangements. Okay, so that's characteristic of staph. Where do we find them? Very commonly, they're just normal microbiota on the skin, upper respiratory tract, mucous membranes like the respiratory tract, for example. Let's see, what else? Characteristics. So they are catalase positive. And hopefully, again, those of you in lab, this should look familiar. It's a very important enzyme, detoxification enzyme. Hydrogen peroxide, those peroxide anions are one of those toxic oxygen compounds that if organisms are going to respire aerobically or they're going to live around other organisms that are respiring aerobically. They have to have a way to detoxify that hydrogen peroxide. So catalase is one of the enzymes that do that, and you end up with water and oxygen. Balance the equation there. So lab people, you know, we put a little bit of organism on a dry slide, took a colony from a plate, and then added a drop of the hydrogen peroxide. And if it bubbled, we saw that oxygen coming off of it, then that was a positive result that the organism makes that enzyme. Okay, this is the test that we use to tell the difference between the Staphylococcus and the Streptococcus. And other genera as well, but the Staph and Strep are really closely related. So they certainly look alike, are similar when we gram stain them. So it's a nice test to differentiate between them. And there's no incubating, we get the results back right away. Some of our staphs will form capsules or at least some kind of slime layer. We talked about how that's very common in the microbial world. Lots of bacteria will do that because there's so many advantages to the organism. They're pretty flexible in terms of their metabolism, facultative, anaerobes. So they respire aerobically and can either ferment or also respire anaerobically. And in general, a little bit more environmentally, tough. I'll put that in parentheses. They don't form endospores. They don't have this super impermeable cell wall like an acid-fast cell wall, but they tend to just persist a little bit better. They're very easy to grow in lab. We don't have to coax them into growing at all. They tolerate salt pretty well, and that's not too surprising. Remember, our skin can be kind of a salty place, and if the staph live on our skin, then that's not too surprising. They tolerate temperature fluctuations pretty well, pH fluctuations. So they're just a little bit more environmentally tough, especially when we compare them to the Streptococcus. The streps are wimpy. Let's zero in on one organism, Staphylococcus aureus. Okay, aureus, this is Greek for gold. Okay, so the colonies often look a little bit yellow. Now, the strain that we use in lab, that's not obvious. We've done that on purpose because we don't want it to be too easy to identify when you're doing your unknown. But kind of creamy, a little bit kind of on the yellow side. If we think about where this organism kind of lives and how it's transmitted, that's what we're talking about epidemiology. It's actually common for it to be normal microbiota. And that's kind of surprising because when we talk about the number of infections that this organism causes, that might be kind of surprising. But sometimes it's considered a true pathogen. Sometimes we consider it an opportunistic pathogen. Scientists argue about how to designate it because the infectious dose is more than 1 million cells at the right portal of entry. So it's a pretty big infectious dose. So for that reason, sometimes people argue it should be called an opportunist. And more often people will have serious disease if they are maybe immunocompromised in some way. So you could go either way, calling it a true pathogen or an opportunistic pathogen. Pretty commonly transmitted in hospitals. Biofilms can form on implants, for example, or catheters. Oftentimes we have to remove the implants when that happens. But it's also just spread by close contact in the community. So it's spread by droplets. So those are sort of the large droplets when we cough or we sneeze. Again, closer than 6 feet. Okay, when it's on those droplet nuclei, those are the teeny tiny bits of moisture like that we sneeze out, then it can kind of waft around in the air for a while and be considered airborne. So this is not, staph are not considered airborne. That would be really, really unlikely to get sick from somebody further away. So usually, again, close contact, large droplets, but you know, if we're ill maybe, and you know, you're wiping your nose or whatever, and then you're touching things, you know, like doorknobs and things, the staph again will stay viable for quite a while on fomites like that. So we pass it around to each other. Anywhere people are close together, schools, daycares, you know, locker rooms, things like that. It's where we pass it around. Okay. If we look at some of the tools in the toolbox this organism has to be successful in establishing an infection in us. A couple of structural virulence factors. I mentioned the capsule, either a true capsule or a slime layer. This is a transmission electron micrograph. I believe that kind of this light gray area is supposed to show the capsule. Remember the functions of a capsule: it inhibits phagocytosis by neutrophils, white blood cells, makes it harder for them to phagocyze the bacteria, and it helps them stick to artificial surfaces. So again, catheters, implants, things like that, helps them form biofilms. Another structural virulence factor, protein A. So we call it structural because it's on the outside of the bacterial cell. So if you look at this, and again, these are virulence factors for Staph aureus. Not any one strain of Staph aureus probably would have all of these virulence factors, but these are just, again, some of the tools in the toolkit. So here's a Staph aureus cell here, and sometimes it has protein A all on the surface of the cell. And what that protein does is that it binds the FC portion of antibodies. Okay, so remember the antibodies, we looked at the structure of an antibody. The FAB portion, the fragment of antibody binding, is the two arms. And then the stem region, that's the FC region. So here's this protein A in orange here. It's binding the stem region of the antibody. So what it's doing, it's kind of neutralizing the action of the antibody. The business end is the fragment of antibody binding. When antibodies bind to bacterial cells, say this way, then it can clump them together, or it can neutralize binding sites, or act as an opsonin. Okay, when it's bound this way, then the antibody can't do anything. So the analogy that I like, it's if you're gonna catch a snake, you don't come at it the front of it, you grab the snake from behind its head. Okay, so if these are the fangs of the snake, you grab it from behind so it can't bite you. Okay. Let's see. Okay. A number of different exoenzymes can be secreted by Staph aureus. Another one that you learned about in lab, coagulase. Okay, the function of that is that it clots blood. So that is going to help the organism just establish itself, or let's see, establish an infection. Okay, so if it can, maybe a person has a cut and their skin, bacteria, Staph aureus are entering. If it can kind of wall itself off by forming a little clot, then our first responders, our neutrophils, our macrophages can't get there to phagocytize it. So it just kind of protects itself. And then when it multiplies a number, runs out and runs out of nutrients, then it will make another enzyme, staphylococcus, to break out of the clot, okay, and invade deeper. Just another reminder, again, for those of you in lab, here's how we do a coagulase test. We inoculate rabbit plasma, right? We incubate. And then for a positive result here, we look for a little bit of a clot. It's not obvious in this picture, but there's a little bit of a clot there. And that's a positive result. There is one other staph besides Staph aureus that makes coagulase, but it's not very common. So usually we use this to help us identify Staph aureus. Okay, so again, makes sense. The staph that causes all the infection is the one that's able to kind of easier establish itself in the host. Some other exoenzymes, one that we talked about a while ago, hyaluronidase. Remember, hyaluronic acid is that matrix between cells. So hyaluronidase breaks down that matrix, or kind of that glue that holds cells together. Okay, so again, the point is invading deeper into our tissues. Lipase, another exoenzyme that you tested for a while ago in lab. This hydrolyzes the bonds in lipids. Our skin can be kind of an oily place. We have sebum that's secreted around our hair follicles. If a bacteria can break down those oils, then it has a better chance of being able to invade. Okay. And then lastly, some strains can make beta-lactamase. Remember, that's an enzyme that alters the beta-lactam ring of some antibiotics, of beta-lactam antibiotics. Okay, and helps them be resistant to some commonly used antibiotics. Okay. In addition, there are some exotoxins that Staph aureus can make. No endotoxin. Remember, endotoxin is only gram-negatives. It's that lipid A in the outer membrane of gram-negatives cell wall. Exotoxins like hemolysins. Hemolysins lyse red blood cells. And we're going to talk a little bit more about hemolysins in regards to when we get to our streptococcus. But Staph aureus actually will show complete hemolysis of red blood cells when you streak it out on blood agar. So you see that yellowing around the colonies there. You would think that this would be a really bad thing to have an organism that's lysing our red blood cells. We think that it actually doesn't have a huge effect in the host. But when we inoculate the organism in vitro in a lab and test tube in a petri dish, then we see the effect really clearly. But probably less of an effect actually in vivo in our bodies. Some of the streps will make enterotoxin. That's a toxin that acts, we talked about this one before, in our primarily large intestine. Heat stable. The reason I bring that up is, say the food, maybe somebody with a staph infection on their hands was preparing the food, it was served at a meal, and then it was put in the refrigerator. And you would think reheating it later might kill the organism, but it would not deactivate the toxin. So Staph aureus food poisoning is primarily caused by this enterotoxin. Again, not deactivated when you heat it up, and it can cause the nausea and diarrhea. Typically not a serious kind of food poisoning, lasts for maybe a day or so. Exfoliative toxin. This is a toxin that causes the skin cells to separate. Right, so we'll talk about a particular infection that can cause in a minute. And toxic shock syndrome toxin. This is a super antigen. We talked about that last time, and I think a lecture before. Remember, super antigens cause massive non-specific T cell activation. Okay. Lots of cytokines produced that cause an overwhelming inflammation. Okay, so we'll look at some of those symptoms of toxic shock syndrome in a minute. All right, so if we take all those virulence factors, what kind of damage does that do in the host? We'll look at some common infections. So we'll start with fairly innocuous ones, and then they'll get kind of on average more and more severe. Start with our skin infections. So impetigo, this is a common childhood skin infection. It spreads really easily. Little kids are rubbing their faces and touching stuff on other people all the time. So preschools, daycares, things like that. It's kind of a pussy sore, usually shows up on kids' faces. It scabs over and then it opens up again. And it scabs over and it opens up, and it just kind of persists, probably due to virulence factors that Staph aureus has. Really need to treat that with antibiotics so that it doesn't scar. Otherwise, it will persist for months and months. So not fairly benign in that it doesn't usually go beyond the skin. Not much chance that it's going to be anything worse than that. But again, treat it so it doesn't scar. My son has a scar right in the middle of his forehead because I didn't recognize what he had when he was little, and it was a while before I got him to the doctor. I got to look kind of hard for it, but it's there. A little more potential, usually still, pretty innocuous, but a little more potential to be serious, folliculitis. That's an infection of a hair follicle. So there's a healthy hair follicle, and then when there's an infection, often gets a lot of pus produced, pusses, fluid, dead cells, due to the action of lipase. Again, that's kind of an oily place, and again, lipase can kind of break down those oils. So it usually manifests as kind of little pimples on the skin, more common in places where the skin has been irritated. So maybe if the patient has eczema, for example, or maybe just been irritated by shaving, or even just wearing tight clothes, then it's more likely to show up. Usually, again, clears up on its own after a few days, but it has the potential to progress into a little deeper tissues. And then when it does, we might call it a boil or a furuncle. Okay, so these are kind of larger areas of skin are impacted here. A lot of redness, a lot of pus. Okay, again, usually these are still going to heal on their own. If they don't, then that's when you would want to get it looked at. They may need to be drained, may need some antibiotics. Again, if these persist, sometimes those furuncles will spread, and then we call them carbuncles. A common place for this to happen is the back of the neck. I'm not sure why. Maybe that's a spot that we miss when we're washing. I don't know, but that's a common place. So when you have furuncles that are spreading, and then we call them carbuncles, this should be looked at by a doctor. When they start spreading, involving some deeper tissues, again, seek some medical help. They may need to be lanced, probably need some antibiotics to get rid of those. Okay, but and it's more likely to be associated, with some fever. Okay, and then, your book just gives a couple more examples of a furuncle here on the left. And again, this, that's looking pretty nasty, you know, red, pussy. That doesn't look like it's going to heal on, you know, on its own. Okay. Yeah, okay, well, we'll move on. MRSA, then methicillin-resistant Staph aureus. This is a strain of Staph aureus. It's just resistant not only to methicillin, but lots of commonly used antibiotics, not only beta-lactams, in fact. These usually present as skin infections, but if they're hospital acquired, sometimes, again, it's a result of, you know, surgery and they're deep within, maybe on some kind of implants, artificial hip or something like that. So a hospital acquired is usually going to be a little bit more serious than a community acquired MRSA. Your book says that 86% of MRSA cases are hospital acquired. So less often, you know, acquiring them at your health club or in a locker room or whatever. Luckily, the incidence of these infections is decreasing. Less than 3,000 cases in the US per year. For some, we don't really understand why they're decreasing. How do you know if you have MRSA? Probably a doctor might be able to tell. I looked this up. Apparently sometimes when you have an unexplained sore on your skin, you might say, oh, I got a spider bite because I don't know where that came from. Apparently that's really rare, actually, to get a spider bite. But MRSA looks a little bit pussier. Boy, anytime you see, you know, redness like that, pussiness, people tend to disregard skin infections because they don't hurt sometimes. But anytime, again, you have a skin sore that after a few days is not starting to heal, good idea to have it checked out. Maybe you need some antibiotics to get rid of it. Okay, because they have the potential to, again, to go deeper and go systemic. Okay, so what I mean by systemic is involving more than one kind of tissue or multiple organs. So typically, Staph aureus is only going to go systemic in somebody that's, you know, not otherwise healthy, maybe has some other health issues going on. If it gets in the blood, bacteremia or septicemia, I believe those two things describe the same, two words describe the same thing. If bacteria gets in your bloodstream, and then basically it can, you know, infect a lot of different kinds of tissues. It can infect the bone, osteomyelitis. I didn't cite this picture very well, but I remember this was an X-ray of a young Ethiopian girl that had osteomyelitis in her femur. And you can see how misshapen that is. She wouldn't have been able to walk. Somebody with osteomyelitis usually has got to be on intravenous antibiotics for a good long time, maybe a couple months hospital stay to try to resolve that. Adults will more often get osteomyelitis in their vertebrae due to the specifics of blood flow to the vertebrae. I guess that's the most common place that happens. A pneumonia caused by Staph aureus is particularly deadly. Again, really high fatality rate there. Endocarditis is an infection of the heart valves. That's pretty serious, obviously. Meningitis, the meninges, that's what covers the brain and the spinal cord, a protective layer. So again, any kind of, we're going to talk about several other kinds of organisms that can cause bacterial meningitis as well, but that's, again, pretty serious. There are a few Staph aureus infections that are considered toxigenic, and that means that the effects are due to toxins produced by Staph aureus. Scalded skin syndrome, that's due to strains that can produce that exfoliative toxin that makes our skin cells basically disintegrate from each other. You can see how patients would be really open to a lot of secondary infections, so infections that are basically come about due to a primary infection. You lose your first line of defense there, your skin, you're open to all kinds of other things. This is one of those infections, like a lot of infections, it's worse in kids, but this one, kids usually will come back from it pretty quick. In adults have a lot harder time for some reason clearing this toxin from the kidneys. Their kidneys tend to shut down. It can be a much more serious an infection in adults. Again, the enterotoxin causes food poisoning. A lot of food poisonings, the organism has to kind of grow up in our body and reproduce. So you might not feel the effects for maybe 2-3 days, even a week. With Staph aureus food poisoning, it's because of that toxin. You can eat it and man, sometimes right away you start feeling bad. 30 minutes it hits you because again, it's just that toxin that's acting. Toxic shock syndrome. So that caused by that toxic shock syndrome toxin, TSST, that we talked about. It's a super antigen. Cases of toxic shock syndrome really went through the roof in about 1980. And at that time, that's a time where a tampon went on the market called Rely tampons that were marketed to be super absorbent. They were basically made out of diaper material. And they told women, you can keep these in for 8 to 10 hours and they won't leak. It's the greatest thing ever. Well, they didn't leak, but that provided just an ideal breeding ground, bloody warm place, changes the pH of the vagina, ideal breeding ground for Staph aureus. So we saw a lot of cases of this toxic shock syndrome. After we figured it out and the FDA pulled that tampon from the market, incidence has gone way down. We still associate this in females with improperly using tampons. So you shouldn't be using a tampon for all nights. You should leave it in for 4 or 5 hours and then change it out. That's the proper way, the safe way to use tampons. So don't leave them in for longer than that. Men can get this infection too. It's usually as a result of some trauma or surgery. The incidence now is quite low. So this is rare. Again, that Staph aureus will be able to make this toxin, but we see a sudden onset, high temperatures, like 104, 105 temperatures, vomiting, diarrhea, a rash that's supposed to look like a sunburn. I don't know, it doesn't really look like sunburn to me, but that's how it's described sometimes. Mental confusion even. So patients can go into shock. So much inflammation, so much T-cell activation that their blood pressure goes way down. And so this is a potentially fatal infection. Okay, so again, thankfully pretty rare. So that's it for Staph aureus. Just want to mention a couple of the other more common staphs, the ones that you hear about all the time, Staph epidermidis and Staph saprophyticus. Staph epidermidis, really normal microbiota, commonly on our skin. Probably, remember we talked about microbial antagonism. Probably it outcompetes other kinds of microbes that would be more likely to cause an infection. So probably usually considered a mutualistic relationship that we have with Staph epidermidis. However, if it gets somewhere in the body that it doesn't belong, it infects a wound, for example, gets in the bloodstream, the thing that you always hear about that this organism can cause is the endocarditis, the infection of the heart valves. Again, that's pretty rare without some kind of other trauma. And then Staph saprophyticus is, again, normal microbiota. Vaginally is where sometimes it causes problems. There's potential to cause urinary tract infections, more likely in women between the ages of 18 and 38. Why younger women? Because usually when women are older than that, they've already had an infection by Strep agalactiae and they'll have some resistance to it. So again, one of the more common causes of urinary tract infections. Lots of organisms can cause urinary tract infections. Okay, lab people, what's the test that enables you to tell these two organisms apart? ANOVA bias and DISC, right. Okay, so a simple antibiotic disc test there. All right, any questions about staph and Staphylococcus, Staph aureus, anything? Sometimes when you get a Staph aureus infection like MRSA or something like that, then you are more likely to get it again. I've talked to students that have recurring staph infections. Okay. All right, well, let's move on to the Streptococcus. Again, will not be covered on Friday's exam. Again, lots and lots of different strep species. I think there's somewhere around 40 or so species. They decided in the 80s that this genus was too big, and they kind of moved the Enterococcus and the Lactococcus. They decided that those were different enough, and they called them by another genus. These are gram-positive cocci in chains. Okay, so we especially will see that chaining when we take our cells from a broth. Okay, you don't always see it if you are gram staining just from a colony. So that's why we like for you to do gram stains from your broth. And if you see these long chains, that's a big tip that you've got a strep. If you don't see chains, don't rule it out, but again, they just might not show up, might not be a culture that's been growing long enough. Okay, they are non-motile and non-spore formers. And both of those, I just didn't mention that, but that's like staph as well. Remember, what makes an organism motile is whether it has flagella or not. Okay, so we rarely see this around cell with a flagella. It's just, there may be some exception, but usually cocci don't have flagella. There may be some exceptions to that. So they're non-motile. They're just gonna kind of Brownian motion, be floating through a medium. And spore formers are usually rod-shaped organisms. There may be an exception there as well, but okay, so non-spore formers, they are either facultative anaerobes or anaerobes, true anaerobes. There's some variation within the genus. All the ones that we work with in lab are facultative. It's too hard to work with a strict anaerobe in a beginning microbiology lab. On a related note, some of them are capnophiles. And we might have talked about that earlier in the semester, but that just means they grow best in an increased concentration of carbon dioxide. Okay, so when you put brackets around something, that means concentration, so increased carbon dioxide. So because some of them are true capnophiles, there are a few of them that have to have elevated CO2, higher than atmospheric concentrations of CO2 in order to grow. Some of them will just grow better that way. So that's why in lab, we incubated our organisms in a candle jar. Okay, you put them in a big jar. You light a little votive candle in there, put the lid on. The candle will go out when it runs out of oxygen. And the oxygen's not the important part. The important part is we get CO2 as a result of that combustion, that lit candle. So a little bit, then we can provide a little higher concentration of CO2. Okay, these organisms are catalase negative. Again, that's a nice way to tell our staph apart from our streps. They actually make another enzyme called peroxidase that does the same thing, that detoxifies that hydrogen peroxide. It goes about it a little different way. It uses that electron carrier, NADH, but you'll see that we don't end up with any gas being produced, no oxygen being produced. So you're not going to see any bubbles come off of that organism when you add the hydrogen peroxide on a slide. Okay, so there's a couple of different ways that we will classify the Streptococcus. The way that we rely on in lab primarily is by their hemolysis patterns. So when we streak them out on blood agar, we see three distinct ways that the organism might break down those red blood cells. Our alpha hemolytics, here alpha, we get kind of a greenish color change because they're only partially lysing the red blood cells. The hemoglobin stays intact, so you get kind of a greenish color change there. Total red blood cell hemolysis are beta organisms that do beta hemolysis. You get the kind of a yellow clearing. You can see right through the media now, okay, because again, those red blood cells totally lysed. And then this is supposed to look like a gamma. It's kind of hard to see that there, but there are organisms, streps that don't lyse the red blood cells at all. So we just call it gamma or no hemolysis. Okay. So again, we've got to streak them out on blood agar. Some of our streps don't grow very well unless we put them on blood agar. It's a really rich medium. Again, they're a little bit particular. But the other way that we like to put our streps into categories, we do it by hemolysis pattern, but we also do it by Lancefield groups. And this is a grouping system that Rebecca Lancefield came up with in 1920. So 100 years ago, she noticed that there were different carbohydrate surface antigens on the surface of streps. So if you can picture these carbohydrates on the surface that act as antigen, we can make antibodies that will recognize one carbohydrate from another. Okay, usually carbohydrates aren't very good antigens. They don't elicit very strong immune response, but she realized that you can type this out. So not to memorize this, just to show you that, when you hear the term, oh, group A strep, that refers to these carbohydrates on the surface of the cell. For some of these groups, like group A, it's only the species Strep pyogenes. That's the only species that's group A. But for group C, you can see there's several different species there. So again, not to memorize, it's just a way that we tell, it's like a taxonomic thing for streps. The problem is there are some streps that don't have those surface antigens. So we call them non-Lancefield streptococci. Okay, so to me, it's not a perfect system, but one that you see and you read about and kind of want to know what that means. All right, so let's zero in a little bit on Strep pyogenes. We'll talk the most about this organism. It causes all kinds of different things. It's beta hemolytic group A. So sometimes you see the abbreviation GABHS. I think your lab manual maybe uses that. That just stands for group A beta hemolytic strep. So sometimes we call strep pyogenes this GABHS. Okay. Where do we find strep pyogenes? It can be normal microbiota in the human throat, maybe transiently. So sometimes that will kind of complicate when you're trying to do a strep test for somebody that has strep throat. Well, you might have some normal flora in there. How do you differentiate? Sometimes on the skin, it's less commonly normal microbiota than some of the staph. Respiratory secretions. So again, large droplets, coughing, sneezing, within closer than 6 feet. There are streptococcus that animals have. They have their own streptococcus, and usually they don't cross species. So animal strep doesn't usually infect humans, but it has been documented. So we'll just say zoonotic transmission rare. But again, they have documented cases of somebody getting like strep throat from their dog or something like that. And this slide needed a picture, so I included this one. There's a website called giantmicrobes.com, and you can go on there and get little plush, you know, whatever you want, Zika or Ebola or anything. They've got all kinds of cool stuff. So this is the Streptococcus. So, you know, here, 2 round cells chained together. There's a fork and a knife on him because strep pyogenes causes flesh-eating disease. So, I don't know. Anyway, he's kind of cute. Although flesh-eating disease would not be. Don't want to minimize that. Okay, so let's think about some virulence factors here for strep pyogenes. Again, a couple structural ones. Most often, the strep pyogenes is going to make some kind of a capsule, and oftentimes this organism, that capsule, is this particular polysaccharide called hyaluronic acid. So that is a compound that we have in our bodies. So it's kind of a dirty way to hide itself. We're not going to make antibodies against something, hopefully, that's similar to our own self. So it kind of evades phagocytosis because we don't have a good immune response to that because it's similar to something that we have. And strep pyogenes have this virulence factor. We mentioned this in the past when we were talking about first introducing kinds of virulence factors. M protein is an anti-phagocytic factor. And you can see here's a strep that has the M protein and here's one that doesn't. They typically cannot establish an infection unless they have that M protein. And I'm not sure what M stands for. I think it might be motif, surface motif. But what it does, remember we talked about there's 3 different ways that complement can get activated. Sometimes with the classical activation, it's an antibody bound to an antigen that causes C1, then to start the complement cascade. Sometimes complement is activated by lectin binding to a monosaccharide called mannose on the surface of bacteria. The third way is that alternative pathway where C3 is just kind of randomly splitting on its own. It will split much more if it's in the neighborhood of a pathogen, as well as there's these proteins factors in the neighborhood that we didn't really talk about. But the M protein will inhibit some of these protein factors involved with that alternative activation of complement. So again, remember that complement is a really potent opsonin. So where some of those like C3b, C5b would bind here and then be more likely to be phagocytized, now those complement fragments are much less likely to be there. So it's going to resist phagocytosis, again, not just by the capsule, but because of this M protein. There are actually 2 different well, there might be more than 2, but there's 2 main variations of M protein. There's a rheumatogenic M protein, and we're going to talk about the repercussions of this. We can have some autoimmune repercussions. That's where our antibody that we make eventually to target this organism cross-reacts typically with heart antigens. And the other kind of M protein, the nephratogenic, we make our antibodies cross-react with kidney antigens. Okay, so we'll come back to that because that's going to potentially cause some complications of a strep infection, some autoimmune complications. Okay, we'll start next time, get into some more of those exoenzyme virulence factors. Have a good day. See me if you have questions. Got office hours tomorrow.